Good day, everyone. I am Eva May V. Alahas, and I am here to present the sexual communication in the Drosophila genus. The Drosophila genus is composed of a wide range of species, some of which are genetically close, living in different environments in allopatry or asympatry. Here are the objectives. In insects, sexual behavior depends on chemical and non-chemical cues that might play an important role in sexual isolation. In this presentation, it would tackle about the current knowledge of sexual behavior in the Drosophila genus. Drosophila courtship behavior. The courtship ritual is composed of stereotyped but nonlinear steps that constitute a true dialogue between the partners. So here, as you can see at the illustration below, here are some of the courtship choreography. The orienting. When a male encounters a potential female mate, he will orient towards her and taps her abdomen with one of his front legs. This allows him to perceive her pheromonal characteristics. If he decides to pursue courtship, he will follow her, extends the wing that is closest to her, and produces a love song. He will then lick her genitalia with his proboscis and attempt to copulate by bending his abdomen. The female transmits acceptance or rejection signals to the male throughout courtship. Signals emitted during courtship have characteristics which are to species specific or unique to a given population and are import for intraspecific recognition and reproductive success. Non-chemical stimuli. There are two kinds of non-chemical stimuli, the acoustic stimuli and the visual stimuli. Acoustic stimuli. During courtship, males of most Drosophila species emit sounds by vibrating one or both wings. The courtship songs are very diverse in the genus Drosophila, and their acoustic characteristics are specific to species in the Melanogaster subgroup. The Melanogaster males produce two songs. The first one is the pulse song, which is the series of rattles produced by short wing vibration, and the second one is the sign song, which is the burst of humming produced by longer wing vibration. The pulse song and in particular IPI are critical parameters for interspecies recognition. Visual stimuli can be dynamic, movements or locomotion, or static, pigmentation, color, form, and vary quantitatively and qualitatively within the Drosophila genus. In some Drosophila genus, these signals can also modulate female choice. Sex pheromone signals. The first one is the cuticular hydrocarbon generalities. In the Drosophila group, it acts as recognition signals and sex pheromones. They are perceived at a short distance by olfactory organs on the head. The highest variation, particularly in those involving CHE sex dimorphism, occur in the Melanogaster subgroup. The second one is the pheromonal role of CHEs. In Drosophila, some CHEs, generally the most abundant, act as sex pheromones during courtship behavior and mating. The high variability in CHE numbers and their amounts could serve to improve attractiveness of potential mates from the same species or population. The third one is the pheromonal rule of CHEs from immature flies. Homosexual behavior has been described in Drosophila. Mature Melanogaster males vigorously court immature males. Immature flies contain complex CHEs mixture with 23 to 37 carbones. Most of them are methyl branch or mono or dino unsaturated. This particular unsaturated CHEs could explain the attractiveness of young flies, males or females, to mature males. Role of CHCs in reproductive isolation. Role of female pheromones in D. melanogaster reproductive isolation. CHG profiles can vary within species, and more studies report non-random mating between divergent populations. In this species, as CHG polymorphism linked to the geographic origin of population results in the production of different female sex pheromones. The gene involved in this female pheromone polymorphism has been characterized. Nevertheless, the exact rule in reproductive isolation has yet to be clarified. Role of male pheromone in D. melanogaster reproductive isolation. In D. melanogaster, two studies suggest that male pheromones play a role in reproductive isolation between population. There is a geographic CHG polymorphism concerning the males. Cosmopolitan males synthesize large quantities of 7T, contrary to males living in hotter areas which has high and low quantities of 7P and 7T respectively. Other parameters involving the male, such as morphology, courtship behavior, or pheromones, might also be responsible for this isolation. Plasticity and evolution of CHCs. Plasticity of hydrocarbons. Several factors may affect hydrocarbon profiles, notably temperature, food, and social context. In other Drosophila species, selection for resistance to desiccation leads to a modification of hydrocarbon profiles, which is generally characterized by an increase in chain length. Long-chain CHCs have higher melting points, which gives them a superior capacity to limit water loss compared to short-chain CHCs. The next one is the evolution of CHC profiles. Natural and sexual selection can play a role in CHC evolution. Several studies have shown that natural and sexual selection can influence CHC evolution and male reproductive success. Non-hydrocarbon pheromones. Production of cis-vaccinyl acetate. All species of the D. melanogaster group produces cis-vaccinyl acetate. This ester is exclusively synthesized in the male ejaculatory bulb. However, trace amounts of CVA were found on the cuticle of virgin females based on direct ultraviolet laser desorption. Role of CVA in mating behavior. A significant amount of CVA present in the male is transferred to the female during mating along with the seminal fluids. CVA has been proved to act as an anti-aphrodisiac pheromone, preventing males mating newly inseminated females, but also interactions between males. Role of CVA as an aggregation pheromone. The CVA transferred to the female during mating is deposited onto the rearing medium within 6 hours after mating completion and attracts both sexes. There is a specific CVA needed for their aggregation effect. It acts in synergy with food odors to attract other flies. So, other behavioral rules of CVA. When population density is low, CVA is present at a relative low concentration and acts as an aggregation pheromone, resulting in increased number of flies on the same food source. This highlights a possible role of CVA in controlling population density. So the last one is the polar compounds and triacylglycerides. Recently, the use of non-conventional analytical methods has allowed the characterization of more polar curricular compounds. Direct ultraviolet laser desorption ionization mass spectrometry has been used to analyze cuticular compounds. One of those compounds is a male sex pheromone. It is present at high levels in the male anogenital region from D. melanogaster and transferred to the female during mating. One difficulty for their study is their unconventional structure and their diversity within species. That could be all. Thank you.